Hi everyone, welcome to this series of videos on DNA replication, transcription, and translation. This is a huge amount of information, everyone, and so it's going to be broken up into, I think, 5 videos, maybe 6. And so I tried to make them small chunks so that it's easier for you guys to digest. I do want to share with you, as always, the Sunshine State Standard that goes with this. This is L.16.3, which is to describe the basic process of DNA replication and how it relates to the transmission and conservation of the genetic information. We're also going to be looking at 16.4, 16.5, and 16.9, and I'll try to explain those to you when we get there. One interesting thing about the EOC is that content limits are sometimes explained, and so you may want to read this. Items requiring the analysis of base pairs for gene mutations are limited to genes and changes in a single gene. When we talk about mutations, which isn't necessarily going to be this video, when we talk about mutations, we're not going to be talking about multiple mutations, but a single mutation. So that's not really applicable here, but items may refer to but not will assess the cell cycle, mitosis, and or meiosis. Now we've already covered that in other videos, but you need to know that for the information that's going to be assessed here. I'm going to bring it back up, so I hope you've already watched those videos. You are not going to be required to memorize specific conditions resulting from chromosomal mutations, like certain types of diseases that come from mutated chromosomes, but we may refer to the process of meiosis in the context of mutations, but will not assess meiosis in isolation. So that's a lot of information. As I go through here, I'll try to give you a little bit more on that. We're going to be looking at these types of things more later on. We're going to start out with talking about, we are talking about DNA, and everything we're talking about here is happening at the DNA level. We call this molecular genetics because these A's, C's, G's, T's, these are made of atoms, and so these are a molecule. It is a much smaller level than the chromosome. We can study chromosomes in genetics. Gregor Mendel has a kind of a his inheritance patterns tend to follow moving chromosomes around. We talked about chromosomes and moving them around in the cell cycle and how the chromosomes have to be copied first and then moved properly into each new cell. That's the whole purpose behind mitosis. But we're actually going to zoom in even closer now to the chromosome and look at parts of the chromosome to understand how it works. So let's be very clear about what we're talking about here. We are in the nucleus, and inside of the nucleus, remember, chromosomes can be found in 2 forms. Chromatin, that's with an N, not the D here, that's a whole different structure. We're looking at chromatin, which is when DNA is unpacked and ready to be used. Or if you'll remember, when we go through mitosis, one of the first things that happens is that the chromosome packs up, and that's what we see here, these structures of chromosome all packed up, and they are not able to be used, but they are able to be moved safely. So we rarely, in terms of the cycle of the cell, we don't see chromosomes very often. Mostly it's in the form of chromatin, T-I-N. But I want to tell you that what you're looking at here is packed up DNA. Chromatin is the unpacked DNA, so it kind of looks more like this. It's all kind of fluffed out. But here it's packed up and ready to go. But if you look close at it, you can see that it's DNA that's wound up and wound up and wound up several times. We call these supercoils, and they're wound around these protein structures called histones, because as we wind it around these histones, it gives us a stronger structure, which is the point here, is to take all this DNA and wrap it up into these stronger structures so that they can be moved safely without breaking. If this DNA breaks, it's game over. The cell dies. And the cell doesn't want to die. So winding it up and packing it away is important. But from this point forward, we're not going to be talking at the chromosome level. We're going to be talking about at the molecular level. So we're going to be looking at the DNA which makes up the chromosomes, and we're going to be taking a close look at that. Now, DNA is made up of monomers. It is a polymer. I'd like for you to notice it's a very long chain. If you remember, we talked about polymers. Polymers are long chains of monomers that are put together in a length. And in fact, this is actually two polymers put together. One side, each of these ribbons is a monomer. I keep doing it again. Each of these ribbons is a polymer, and these polymers are stuck together. So this is a double-sided polymer called DNA. Now, remember, polymers are made of monomers. Well, here's four of the monomers. There's actually a fifth one that's found only in RNA. It actually replaces thymine, but we're going to talk about that later on. So the four monomers that make up DNA are guanine, cytosine, adenine, and thymine. You'll notice they have a very similar structure. They have a phosphate group as what we call the backbone. Then there's a sugar called deoxyribose, and then there is a base. Now this base is what is different in all three. I'm sorry, all four. Adenine and guanine are what we call purines. They're larger double ring structures. They have two rings on them. And cytosine and thymine are pyrimidines. They're single ring structures. And these can stick together right here. You see how they bond together at the phosphate group to make a long chain. And they'll go to the left and to the right. Literally billions of them can be stuck together to create the long chain of the DNA polymer. However, uh, this order is not random. This order is specific. And understanding how this was put together, understanding how this structure looks, took a long time. Gregor Mendel studied the rules of inheritance in the late 1800s, and it was not until the 1940s before somebody figured out that this is what held the key to inheritance. This is where the code. And in between that, it was discovered that there is a code, and lots of information about this was discovered. But nobody really understood how the molecule worked until two guys, and I'll show you them in a minute, came along and were able to say, no, this is the structure, this is how it works. And they won a Nobel Prize, and they're some of the most famous scientists of all time. Now, one thing I want to tell you while I'm here is that this is one side of DNA. There's a similar one on the other side. I'm just kind of moving my mouse up here to show you. It would be over here. And there's rules about what an A will bond with. So when I talk about an A bonding to a T, if there's an A right here at the end of this side of the A, and right over here on this side of the T would be flipped around and put over here. You'll see again in a minute. And they bond right there. It's called a weak hydrogen bond. An A will always bond to a T, and a G will always bond to a C. So if I have a G over here, there's always going to be a C over here to make what's called the complementary side. This is the discovery of this, and the understanding of this was what is the genius behind the guys that discovered the DNA shape and the DNA structure. It took a lot of math and a lot of science to do that. It's not like we can see this with a microscope, really. I mean, we kind of can now, but we certainly couldn't back in the 1940s. They figured all this out using chemistry and math. But what we're going to be talking about is how that structure is built, what it means to have two sides, and how that gives us all of our inheritance patterns. So here we go again. Here's the A. Now, we left off all that other stuff because it's all the same, so we don't normally draw that. And in fact, a lot of times we won't even show these shapes. We'll just call A's, G's, C's, and T's. But this A, it was a double ring. See here? And don't worry about the color. They get different because I stole these pictures from different places. But here the A has a double ring right here, okay? And it always bonds to a T. Now, we know this because way back in the 1920s, a guy named Erwin Chargaff discovered this. He took we knew what DNA was. We had found it, when taking blood or other types of stuff. You can actually, there's a pretty simple experiment. You can see it at the house. You can take peas and blend them up and add some soap and do a little bit more blending and then add some alcohol, and next thing you know, you can stick kind of like a toothpick down in there and you can pull out the DNA. It looks kind of like wet cotton. We've been able to do that for a long time, but we never understood how they went together. But Chargaff was able to take some DNA, and he was able to take quite a bit of it and kind of put it in a centrifuge, which spins it at a very high speed, and it separates out, and the heavier ones go to the bottom and the lighter ones go to the top. And I think guanine is slightly heavier than adenine, and cytosine is slightly, I think thymine is slightly heavier than cytosine, I'm not sure, but they separate it out. And what he was able to do was he was actually able to measure, once he had a pile of it, I'm talking like a test tube full, and spin it out, he was able to measure out how much of each there was. And it was different per species. So when he took, like, I don't know, green peas and did it, he got a different amount. And when he did, when he spun out, I don't know, like a rose or something, he probably did tissues from animals too. But when he did this, he noticed that there was a different amount of each at each time. But he did notice something very interesting. The amount of adenine always equal the amount of thymine, and the amount of guanine always equal the amount of cytosine. And this was really important because it means that there's always the same amount of adenine and thymine. And now when the next guys, these guys right here, Watson and Crick, 25 years later, were trying to figure out how this thing go together. They knew that Chargaff, and in fact it's called Chargaff's rules. They knew Chargaff's rules was that adenine had to be the same amount of adenine as thymine and the same amount of guanine as cytosine. And so when they're putting together their models, and they worked at King's College in London, and they had the groundskeeper actually make these pieces for them. They gave him specifications, and he went into the metal shop there and he built this for them, and so they could play with it like Legos or Tinker Toys, because that's really how they did it, and it's really the key to how they figured out one of the keys to how they figured out how it went together. But um, they knew that they had to have, if there was 20 adenines, there had to be 20 thymines. If there was 17 guanines, there had to be 17 cytosines. And that was one of the major rules that helped them to understand how the molecule went together. Another one was this lady. Her name was Rosalind Franklin. I think I have that right. Yeah, Franklin. Rosalind Franklin, she worked next door. She had a lab right next to these guys at King's College. And they were friends. You know, I'm not saying they were like good friends, like they went to each other's house at Christmas, but they were friends. And these two guys were trying to figure it out, and they were trying to understand this, how this went together. And she was too, because they all knew that this was the current prize. I mean, whoever figured out the structure of DNA was going to win recognition for years. You know, they're going to be some of the most famous scientists of all time. These guys were trying to put together a model where she was doing something called X-ray crystallography, which is a really interesting way. She learned how to use an X-ray as kind of like a microscope, kind of like a magnifying glass. But if you understand how X-rays work, when you bounce them off a very small structure, you get a picture of the structure, that if you understand how to read it, you can kind of understand some things about the structure. Remember that we're talking about microscopes smaller than microscopic, okay, so small that the microscopes couldn't see that, and so as a result, we just didn't have a really good idea of how this structure worked. Her X-ray crystallography found out a very key piece of information that these guys didn't have. And so one day, when she went to lunch, these guys, some say they snuck into her office, they say they just went into her office and pulled out a picture of her X-ray crystallography of DNA. And they noticed that it was even all the way down, which meant that whenever you had a purine, you had to have a pyrimidine on the other side. And this told them that there was simply no way that a purine could stick to a purine or a pyrimidine could stick to a pyrimidine. It had to have a purine on one side and a pyrimidine on the other. You add this to Chargaff's rules, which means that there's the same amount of adenine and thymine, and they realized that it had to be that on one side there was a nucleotide on one side and a nucleotide on the other, and if there was a purine on one side, there had to be a pyrimidine on the other. And they went back into their office and they came up with it. And literally that afternoon, they wrote a one-page paper, took it down to the patent office, and had it published. And they became some of the most famous scientists of all time. In fact, this double helix model right here is called the Watson and Crick model, because this is what they came up with, and to this day we know that it still works. They were awarded the Nobel Prize in the late 1940s. They won $1 million. It was great. Franklin actually died a few years right around the time they won the Nobel Prize. She died, and whether it was out of guilt or out of just being good guys, I don't know. But somehow later in life, and she was mad, by the way. When she found out that they got her picture, she was mad. But somehow later in life, they decided to posthumously share the Nobel Prize with her. So if Rosalind Franklin was also given recognition for the Nobel Prize, and somehow they gave her some of the money, I don't know. But as a result of all of that work of Chargaff, and actually going all the way back to Gregor Mendel, but Chargaff, a few other people, and then these guys, including Franklin, Watson is the older guy here, Crick. Watson's long dead, but I saw a picture of Crick a few years ago. He looks terrible, but he's still kicking. She died a long time ago. But it all came together to give us this structure. And this is how theories work. It's not just one person. It's a bunch of work from a bunch of different people. Sometimes they're working at the same time. Sometimes they're years ahead of time. Like Chargaff was around long before these guys, 25 years long before these guys. But they took his work, and even going all the way back to a guy named Gregor Mendel in the late 1800s, and all their work culminated together to help these two guys figure this out. And we now know the structure of the DNA model, and not only that, but how it works. And so, uh, mathematically, chemically, they were able to understand by using this model and a couple other things. They were able to understand not only the structure, but how the structure has to work. And you'll notice that at the top of this line, there's something called a 5 with a little dash. It's called 5 prime and 3 prime. They knew that the two sides kind of headed in opposite directions. And notice over here, they're called anti-parallel strands. Notice over here that the little the little nucleotides kind of are on the top. They're kind of flipped around backwards in these guys. This strand runs this way and this strand runs this way. They're anti-parallel. Now they stick together. You can see that they still together. C bonds with G, A bonds with T. And then when that all gets all bonded up, it kind of naturally twists together to create this double helix. But they are running in different directions. Now this is all I'm really going to talk about this, but if you ever study this, this is really crucial that you understand that these things run backwards. I may mention it one more time when we talk about replication, but it'll just be brief. I just need you to understand that they do run in opposite directions. They're kind of mirror images of each other, okay? All right, how do they stick together right here in the middle? These right here in the middle are these things called weak hydrogen bonds. These stick together. It's kind of like magnetic stuck together. It's kind of like static, really. They stick together. It's strong enough to hold them together until something comes along and breaks that bond. It's not a very strong bond. It's relatively weak. It's way weaker than the other bonds you see here. This is a bond here, this is a bond, this is a bond. These are much stronger bonds than this bond right here. This weak hydrogen bond is just enough to hold it together. Okay. Interesting to know that between the G's and the C's there are 3 bonds, and between the A's and the T's there are only 2. That's a really important thing that keeps the T from bonding to the G and keeps the C from bonding to the A. So sometimes that mistake happens, by the way, but for the most part, this is how we keep the right things lined up. Notice that a T and a C have a similar shape and a G and an A have a similar shape. How come the T doesn't accidentally bond to the G? It's because there are 3 bonding sites on the G's and the C's and only 2 on the A's and the T's, if you'll notice that. These weak hydrogen bonds are easy to make and easy to break, and that is crucial because it allows us to do some really crazy things with this DNA molecule. We're able to literally, we call it unzip it, just like a zipper. If you ever watch how a zipper works, something's going to come along here in a few minutes. We're going to talk about this. It's called an enzyme, and it will actually break these bonds and let this thing split apart. And then sometimes we have to put it back together, so another enzyme comes along and puts it back together. It's just like a zipper. But making and then breaking these bonds is very simple. So the base pairing rules are very simple. A G always bonds with a C, and an A always bonds with a T. And it works that way every time. Now I can have a T on one side or a T on the other, and that T bonds with the A. If there's a T over here, there darn sure better be an A over here, or else we got a mutation, which can be a problem. Most mutations are bad. Now, this then gives us a very long chain of these nucleotides, and they're not random. They're very specific. And when we put these nucleotides together in a specific order, we get our genetic code. This is the code. The order of these nucleotides is the code, and that code is what makes us us. Okay, you inherited a unique code from your parents, and that made you uniquely you. Unless you're an identical twin, there has never been a genetic code exactly like yours in all of history. Just the odds of that happening are so unbelievably low. It's never happened. So you're very unique, and it's because this code inside of you, and it extends literally. Here's 4 of them. See these 4 right here? There's literally 3 billion that make you up. So there's a lot of code, and that code gives you all of your traits. Okay. Watson and Crick figured this out by using Chargaff's rules, and the fact that whenever he spun those out, there was always the same amount of guanine and cytosine every time, and there was always the same amount of adenine and thymine every time. Chargaff knew that, and that's how Watson and Crick were able to know that there had to be an even amount of guanine and cytosine, and that meant that basically if there was one on one side, there was a complementary one on the other. By the way, we call this complementary. This strand has the complementary strand over here. If I were to black out this side right here and only give you the one strand, because of Chargaff's rules, you should be able to tell me what's on the other side. If right here where I'm pointing, if there was an A right here, you should be able to instinctually know that over here on this side is a T. Very good. Okay. All right. That is the end of the structure of the DNA molecule. We're going to go into a few more things here in just a minute or on the next video, but I want you guys to have this one in the bag. So if you need to rewatch this, don't forget that there is a quiz that goes with this, and I'd really like for you guys to take that quiz. All right, I'm going to stop it here, and this is the end of video 1.